Welcome to the Grand Plaza tonight. I think it's still January, so I'm allowed to say Happy New Year to everyone. Nice to see so many people back in the room, and we shouldn't forget to say hello to the people further afield watching online. Really excited about tonight's talk. Tonight we have Dr. Chris Holland from Sheffield University, and he's going to talk to us about silk. So Chris has been in Sheffield for about 10 years now, I understand. He's head of natural materials at the University of Sheffield, and he's also the chair of the natural materials group within the Institute Materials. Prior to Sheffield, he spent 12 years in Oxford, undergraduate, postgraduate, and PhD on the rheology of silk, and then some time as a researcher. So he's well versed in the in the field. He doesn't quite have 400 million years of R&D experience, but that's what he's up against in trying to make synthetic versions of a very well designed natural material. So he's going to speak to us this evening about silk, and as you find out, it's a hugely adaptable material used in a wide variety of applications, and it's the challenge to take that inspiration from the natural world and develop that into high performance and sustainable modern materials.
Super, thank you so much.
First off, good evening, everybody. It's a real treat to be here. I'm going to talk at you, or with you, hopefully, for the next sort of 45, 50 minutes or so. What I really want to do is just share a bit of my enthusiasm. I want to share a bit of enthusiasm for a material that I've been working with now for, crikey, coming up to 18 years. It was my master's project. I started back in 2004.
So it really is a treat for me to talk to you today. I'm going to share a bit of enthusiasm about an example of a natural material, which we'll get on to in a second. This is my group. I've been in Sheffield, as I've been very kindly sort of introduced by Mark. The Natural Materials Group is this brilliant combination of people with a range of different backgrounds. We have physicists, we have chemists, we have mathematicians, bioengineers, biotechnologists, all working together in a relatively sort of Keith Robinson style room in the Hadfield building up the hill. I like it because I'm a firm believer, even though I'm a biologist by training, I'm a firm believer that if you build a piece of equipment, you understand the test it's doing on it. And so that's where sort of the engineering me has sort of grown over the past decade. And so I'm a builder. We have a range of different types of bits of materials testing kit. So standard tensile testers. We have rheometers. I'm going to expose you to quite a bit of rheology today, but also standard characterization equipment. But what we use this for is that we use material science to ask biological questions. We learn about the natural world, and in return, it very kindly gives us inspiration. It gives us blueprints, solutions to problems that we are facing, that we can take inspiration from. So that's our research group. Our research areas, I'm going to talk today about silk and how this is basically the world's most advanced polymer pultrusion factory, which has solved all of our green and recyclable plastics problems. But it's not just silk that we look at, because we have sort of an expertise and capability in the group testing soft and squishy things. We're very interested and in the past have tested rotator cuff tissue. We've tested the difference between horses and cows, the equivalent of joggers and couch potatoes and their knees, understand the sponge, the cartilage inside there. We've looked at how plants heal themselves, the latex that they produce. Your poinsettia is actually a euphorbia, which in Africa would be mistakenly called a cactus. Actually, go fishing with that. Really interesting stuff. They take cactuses in Africa, and that latex is super poisonous to koi. If that falls in your pond, it will kill all of your fish. But they use it for fishing. They'll take what looks like a cactus, throw it in the river, and all the fish will float up a little bit further down. But that's an anti-predatory mechanism. It is a healing mechanism as well. It's a glue that binds to stop the plant from leaking. We also have a capability and interest in the group for looking at gastropod mucus. This is snail and slugs and the mucus that they produce, which can be this fascinating adhesive, or it can be a lubricant. That's some of the work that you may have seen from Lauren, my PhD student, earlier on in this year, or previous year. We've asked questions about the difference between spitting and non-spitting cobras, and if the flow properties of the venom is optimized, the fang design. We fortunately don't have dangerous snakes in the lab. We had some friends in the Liverpool School of Tropical Medicine do the snake wrangling for us, and then somebody jumping on the train over from Liverpool as we tested the flow properties of its venom. And then we've got some work on hair as well, originally starting out this interesting beast, the African crested rat, which has holes in its hair. Those holes that you may see are a specific adaptation to this animal. This animal also likes eating the bark, or specifically the root, of the poison arrow tree. The poison from the poison arrow tree is in the bark, it mashes it all up as saliva, and then licks these holy hairs, sequestering poison as an example of a mammal that actually has poison. It's a very, very rare thing. And so all of these have specific adaptations. We also have some really interesting work at the moment looking at human hair. So that's Elle, my PhD student, who's here tonight, looking at damage in hair. And altogether, these are materials used outside of the body. And so they have a lot of specializations that we can learn from because we use materials outside of our body. And the prime example I'm going to be giving today is looking at this. The marvel of nature. This material is made on demand. The speed at which you can pull silk out of the spider is being transformed into one of the world's most impressive materials, a toughness that can't be matched and a strength that's incredibly impressive. What that means, the spider is able to encapsulate its prey there to stop it wiggling around. But what this has to do with this problem is what I will be touching on a little bit today, because we do have a global plastics problem. We had a global plastic problem, then we had a world pandemic, and that sort of like flew off the radar a little bit, to be perfectly honest. But the amount of plastic pollution that we now have as a result of PPE that people have used is still going to be a continuing problem. So finding new ways to produce plastics in a green, sustainable and biodegradable manner with a low energy input is one of the motivations that we have for looking at how silk is spun. And that's one of the key differences between silk and other biological materials. If we were to look at hair, bone or feathers, these are marvelous large scale structures, but they don't appear overnight. You know, these structures take years, months and years to grow this large. Whereas looking back at our spider, these materials are made on demand. And we actually define silks by their material processing. Silks are spun. So looking at how silks are spun and how we go about spinning our materials is the sort of focus of today's talk. Now, I'm not the first person to look at how silk is spun. I sort of stand on the shoulders of previous giants. So the first being this fantastic chap called Termeyer, who, this is one of the world's oldest patents, actually. They wanted to see if they could commercialize spider silk production. Termeyer was a refugee and ended up in Milan and wanted to try and create the finest silk possible by looking at spider silk. Had a house filled with rotting meat, actually, that would generate flies and spiders that would just be in the rafters that he would then sort of collect and reel onto this reeling device, then taking that silk and trying to create a garment. Now, unfortunately, one of the problems with this is that you don't get much silk out of a spider. He only managed to make a few sort of very specialist garments that he basically gave to kings and queens in order to sort of ingratiate himself in those social circles. In the end, this didn't actually work. It didn't work very well because he wasn't able to produce enough of it. And he ended up reverting back to using cocoon silk, which you could gather up a lot of and turn into a garment. But this is inspiration for some of the questions that we can ask, because a spider always produces silk behind it. It always leaves a trail. It's one of the reasons why if you have a spider on you and it falls off, it will hang. That is known as its lifeline, its dragline silk. The reason why it produces that is obviously for protection, but we can use this as a mechanism to explore natural silk production. We can take a spider, restrain it gently, far gentlier, that's a word, in a more gentle manner than Termite did. We will restrain it and we can pull silk out of the back of it. The spider just thinks it's going for a walk, but we can control the speed at which that fiber is produced. We can control the temperature at which that fiber is produced, because these are cold-blooded animals. We can control the humidity in which that fiber is produced. So we have access to this amazing experimental system where we can really look at how processing affects the performance of a biological material. So here's an example of our spider. You'll see. I always pop this in because some people get worried that I hurt the spiders. The spiders are far too expensive and I care about them far too much to upset them. They all get a fly at the end of it. So it's bad news for the flies, but we can reel the spider. The spider, there she is. These are just restraining pins for television, unfortunately. So no harm to the spider, but we're interested in what's happening inside here, because spider silk isn't reeled up like a fire hose inside the animal, just pulled out. It is stored as a liquid jelly, and that liquid jelly transforms into the world's most impressive fiber within the milliseconds that you see, the speed at which it can be pulled out from that body. That is quite similar to the way in which we spin polymers from a liquid or a molten state into a solid fiber, but there are plenty of optimizations. If we are to compare the material properties of spider silk, it is sitting in a material space in a region that we can't quite access, this beautiful combination of both strength and area under curve, toughness. Nature is very good at making both strong and tough materials, whereas industrially we tend to have the compromise. We can have a tough material or a strong material, but we can't often have both. But on top of that, I put a shaded region in here, because by changing the conditions in which that silk is spun, changing the speed it's drawn from the animal, changing the temperature, changing a little bit about the humidity, we can take the same fiber, spin it in different conditions, and get properties that anywhere within this processing space. We're not changing the base chemistry. We may be changing the structures that develop, so much like metals in terms of the way in which you are able to make different grain structures and sort of essentially thermally process. The spider is doing similar things, but from a flow processing perspective. So key properties of specifically polymers from an industrial perspective, and silks here, they're high performance, they're water-based and 1,000 times more efficient than our commercial polymers at solidifying. But I'm not going to lie, they're not there in price. It is very, very expensive to get artificial silks. Mulberry silk, the silk that you have in your ties, for example, is known as the queen of textiles, and that sort of royalistic adornment is specifically because it's very labor intensive, very expensive. World's oldest commercial fiber, silk, originally traded by guards on the edges of the Chinese empire as a currency. They've given little pieces of silk that they could sort of trade with people. But it was a closely guarded industrial secret. When asked, where does this amazing material come from? They were told to say that it was from sheep's wool sprinkled with rainwater and left in the sunshine. And it wasn't until one of the very first examples of industrial espionage, where, depending on where you read it, it was either a monk or a princess, took silkworm eggs in a staff and smuggled them out to Japan. And they were able to sort of start to let the world know about this amazing material. So silks aren't just made by spiders. I sort of touched on that earlier. They're made by silkworms, caterpillars, in order to make a protective cocoon as they undergo metamorphosis into a moth or a butterfly. They are used for hunting, by spiders as they lay down their foraging pattern in the middle of the night. They're for dispersal for their young as they can balloon up into the jet stream using their silk as a parachute. Bees will make silk for the caps of their honeycombs. Lacewings will make it to protect their eggs, and even ants will use it to protect their young and to protect themselves. So silk has lots and lots of different innovations, different uses, and has appeared at least 20 times independently in nature. That innovation and nature circling around on solution, i.e. making a fiber for use outside of your body, has at least 20 times appeared in the insects. That's usually a very good sign that it's something useful. So when I talk about independent evolution, it's like our eyes are different to insect eyes, are different to octopus eyes, but they still perform the same function, collecting light and giving us sensory information, exactly the same as silks. And because it's such a broad church of what a silk is and what a silk does, it's very difficult to define what actually is a silk. But we've tried, and we would say that they are structural proteins, they are spun into fibers and used outside of the body. So we're going to touch on some of this to go a little bit more into the structure of silk and what it's used for. So the structure of silk. I'm happy to take sort of questions at the end. If anyone has a burning question, you're more than welcome to ask at your end. I'm going to dive into the structure in a bit of a general way at the start, but silks are polymers, so that means they have repeated units of very simple sort of backbone monomers. Its primary sequence has always been quite difficult for us to understand because you can't see the wood for the trees because it's so repetitive, lots of glycines and alanines. That repetitive structure, you can start to think of studs on a Lego brick. If you're thinking about studs on a Lego brick, that's a very long piece. You can start to get towards this idea of a secondary structure, and those bits can fold back on themselves. We call this a string of pearls orientation, where those repetitive bits become almost like little nanocrystallites with flexible linkers, but those little crystallites can stack on top of each other. The studs of the Lego bricks can snap into other portions and they can generate this nanostructure. And we're moving up in the length scales here from the atom right up to its nanoscale structure. And then even those little fibrils can join. This is actually data from my master's project. It reminds me how I got into this. This is a birefringence image, a polarizing light image of a piece of spider silk. And what this is showing is a huge amount of orientation along the fiber. All of those chains are oriented along the fiber. If we were to compare this to high performance polymers, Kevlar, PBO, you find that they are incredibly oriented. And orientation can give you desirable mechanical properties in the direction of loading, which is exactly what the spider wants. And if we were to sort of dissect a spider silk, and if I want to ask for money from a granting agency and pretend that I'm smarter than I actually am, I would use grand buzzword bingo and say that silks are multi-scale hierarchical nanostructured biopolymers. But really, they're just absolutely fascinating. They have layers upon layers upon layers of a structural hierarchy that forms itself. It isn't grown, it instantly transforms into nanofibrils that turn into microfibrils that have a skin and a core and bundles. And that structural hierarchy allows for communication and dissipation of energy over different length scales. That's partly why silk is so tough. It has fracture mechanisms inside it, the fracture mitigation mechanisms inside it, has a huge degree of orientation. It is able to have chains that slightly slip over one another but are knotted like a rope, so they don't slip too far, so they can dissipate the sort of tensile stress as a kinetically. So they're incredibly tough, very impressive materials. So where are we in terms of the state of the art? What do we really sort of like know from a commercial perspective of silk? Normally I talk about this at the end, but I thought I'll pop this in at the beginning just to frame some of the challenges that we have as a field. So silk biomaterials today, we have quite a few companies around the world. And even since sort of DuPont, who fantastically announced in sort of like the mid-90s, spider silk has met its match and ran full-page adverts of a spider hanging down as they were creating an artificial silk. Spent $250 million in the development of it. Didn't work out. Or if you're to look at other companies, Nexia, spider goats. People heard of spider goats? Yes, they managed to create a transgenic line of goats back in the day of Dolly the sheep, one of the very first genetic manipulations of a mammal, a stable line of goats that would express spider silk in their milk. They had this fantastic press conference. They gathered all of the silk experts in the Montreal conference back in 2003, which has gone down in sort of history because they were very, very proud of themselves. And look at this, we've managed to get express MASP2 in spider silk in these goats, which is brilliant. Unfortunately, MASP2 is the stretchy part of silk, not the strong part of silk. So they actually put the wrong thing into the goats. The audience cringed. The company lost $120 million off its stock the next day. And it was front page newspaper that was delivered to every door of the invited people at that silk conference. So it's a bit embarrassing for next year. They do too well. The goats are still alive. There are a few. There's a herd of goats in Utah State University looked after by a fantastic chap called Randy Lewis, who's quite literally a cowboy. And there are a couple in Canada who I think they have Sugar and Spice, I think their names are. They were named after different sort of sweet things. They're in the Canadian Museum of Agriculture, sort of living out their days, treating silk. But none of them have really turned into large-scale commercial products. There have been some sort of basically marketing and proof of concepts. There was the North Face Moon Parker jacket, which I'm going to a little bit later. Adidas have made a future shoe. There's a tie that's made out of synthetic biology. Synbio Silk from from a San Francisco company called Bolt Threads. Silk, which is sort of over on this side, is a German company. You can buy an Omega wristwatch strap or a door pull for a BMW concept car that has a whiff of spider silk in it as well. But none of this, we're not running around at the moment with spider silk bulletproof vests, as everybody was sort of promised and is continually promised. So as a field, we're being a lot more conservative. Unfortunately, there's such great marketing to it. Once the marketeers get hold of it, it's quite hard to sort of dial them back. But the scientists and engineers know exactly where we are, and we're still a little bit far out. And in terms of a commercial, large-scale textile production of a spider-type silk, and that's purely because we don't know enough about it. So for the next sort of half of the talk, we're going to talk a little bit more about the challenges and some of the opportunities that we have when we're trying to recreate silks. So I got one of my PhD students a few years ago, Andreas, to go through every single spider silk or artificial silk paper that's out there and summarize it. And these are the different ways of producing an artificial silk. You can take silk directly from the animal. You could get yeast or bacteria to produce a silk-like protein, or you can take silk that's already been spun and chemically unspin it, and that will give you a goo. And once you've got a goo, you've got two choices of trying to spin a fiber. You can use wet spinning, or you can use dry spinning, and then you can play around with some post-processing in order to improve fiber properties. But one of the key things here is that you have to use industrial-based fiber processing. Now that's good because that aligns with industry and scale up. But when we start to look at the material properties, we can basically, at one time, with a type of fiber, tick one of the property boxes. We can make a silk that's as strong as a spider silk, but it won't be as tough. We can make a silk that's as tough as a spider silk, but won't be as strong. And that's because we can't quite get this match of properties, and understanding this natural spinning basically means we need to understand the process variables in order to control them. So what dials do we need to turn in order to make a silk fiber? Well, the answer is there. We just have to look at the animal. What dials did the animal use in order to process silk? And that's exactly where we look at this. So I said that silk is stored with a protein concentrated goo inside the animal, and it transforms into a fiber. Well, this is very similar to what's happening here. So we've got our egg, it's a translucent protein solution, but we put some energy into it, in this case heat, and it undergoes a phase transition. It goes from a liquid gel into a solid. It also turns white as well. That gives us a lot of information about what's happening to the proteins inside the egg. So what about the proteins inside the silkworm as it goes on its journey from a translucent protein gel into a white solid material? Well, we play around with maybe pH, metal ions, add a bit of heat. There are no heaters inside silkworms. They're cold-blooded, so they must use some other tricks. But ultimately, we're putting in energy. We're undergoing a phase transition. We are moving from a liquid to a solid, which requires the input of energy. And that, for silkworms and spiders, is done in the form of mechanical work. Flow. We actually cause the silk to flow this tapering duct. This is stored in a sac and it transitions through this tapering duct before it issues out of the mouth of the animal. And by pulling the dough through the spinning duct, we cause it to flow, and the proteins absorb energy as they are being stretched. We stretch and stretch those proteins faster than they can relax, and ultimately what happens is that a protective shell of water around that protein is stripped off. A water amide hydrogen bond is removed, the protein changes its shape slightly, and it starts to aggregate. And it aggregates and forms much more stable amide hydrogen bonding, and it falls out of solution. So this is where I have a lot of fun in my group giving talks, because depending on your background, you have to view silk from different perspectives. And I'll use different terminologies and I'll use different languages in terms of how I describe things. But if I'm a biologist and I describe the process by which water is stripped from a protein, it changes shape and precipitates. It falls out of solution. Now, I think for some reason we'll stop sharing my screen.
Yep.
Hybrid working, here we go. Right, so you'd say silks have evolved to be denatured, which actually makes no sense if you're a biologist. But if you're a polymer scientist and you describe the process, if something flows, it changes shape and forms a more stable set of bonds between the polymer chains and changes phase, you'd say this is flow-induced crystallization. So depending on your perspective, you can take different tools and different ideas and apply it to answering questions about a natural system. And that's also what we've done. We are really, really interested in, let's see if I make that zoom bit disappear. No. Well, you miss a little bit of the title, but at least the others can see. What we do in terms of silk rheology and flow processing, for the past sort of 15 years or so, we have looked specifically about how silk flows. We are interested in understanding how that translucent protein solution transforms into a solid fiber. And we use a material science tool, rheology, the study of flow and deformation of materials, to please go for it if there is a wiggling that can make it disappear. Ah, there we go. That's gone. We'll leave it at that. Marvelous. Thank you, Mark. We've used rheology, so standard polymer science characterization of the material properties, the mechanical properties of liquids and gels, applying stresses and strains in order to look at the internal friction of the material and how it responds to flow. But what we've really managed to do is we've worked out how to test it in a consistent manner, and we've tested hundreds and hundreds and hundreds of samples. And with that, we've started to get a better understanding about the distribution of properties. And the distribution of properties, what has happened here? Well, this has suddenly got very interesting, isn't it? I can click through. There we go. Right, that's fine. What we find is that after about 15 years of careful preparation, lots of samples, and a classical polymer science approach to silk has given us some model data, gold standards, and insights into polymer processing. What this really means is we're getting a bit of a better handle on how silk transforms. This is our dictionary. This is some of the phrases and the process by which we think silk is spun, going from intra-protein interactions, so within the chain and then between protein chains, and how they go about forming that structure as they transition from this liquid gel into an aligned, oriented, high-performance fiber. And we use rheology to try and look at each of those events over different length scales. We don't just use rheology, we combine it with polarized light. We combine it with small angle X-ray scattering, infrared, confocal, as well as some extensional rheology as well. And all of that is giving us an understanding about the structures that develop as silk is literally spun and undergoes flow. What we're doing now is we're trying to ask a couple more nuanced questions, but equally important is, what is the energetic cost of silk spinning? And by energetic cost, as in, how much do you have to theoretically heat up the silk in order to get it to solidify? You know, does the spider puff and pant after it's pulled out a few meters of silk, or have to do that all day? And all of that speaks to the energetic cost and the efficiency of fiber processing. One assumes that after 400 million years of R&D in life and death situations, that this would be relative. You know, they're quite fit at that point. They should be able to pull silk out all day, every day, and it not cost too much in terms of energy in order to create that. So we're looking at that, and then we're also looking at the process variables. So we can take a sample of silk, shear it, and understand the properties. But how do we go about making structures? How do we go about changing some of the dials that the animal uses in order to improve the way in which it produces silk? So we'll dive into energetic and talk about the energy input solidification. So our original work, a decade ago now, showed that you basically need to shear it fast enough for long enough in order to cause it to solidify. And 5 years ago, it's not just about how fast and how long you spin it, but also how much force you put into it. So how hard do you mix it in order to cause it to solidify? But what we found is that you need a combination of all three. You need to flow it fast enough and hard enough in order to build up enough energy in the system to stretch those proteins and to get them to accumulate energy and transform from a liquid into a solid. And what we're taking away from here is that we're basically spinning silk. We are subjecting it to an increasing flow rate or shear stress that we can see here. And over time, it will transform from a liquid into a solid. And if we look at how much energy or how much work is required on average for each of the samples, it's hovering around about this 2 to 3 joules per gram. And the work to solidification clusters around that 2.75 joules per gram. Now, we can take silk out of the animal and we can cook it, we can fry it, literally in little pans, DSC pans, actually, and work out the energetic cost of cooking a silk. And when we start to compare this to our DSC work, we find out that the denaturation entropy, the energy work required to spin a silk by heating it up, is about 1.78 joules per gram. So from a biologist perspective, this is the same order of magnitude. So I'm a very happy chap. This is actually incredibly close to one another because it highlights that our ability to look at the flow-based efficiency of silk solidification is very, very similar to the brute force way of getting silk to solidify just by heating it up. So these tools and techniques are relevant. The next step that we're taking is to align these with some of the modeling approaches of some friends we have in New York. For other process variables, typically, if you're a polymer scientist, you say rheology is entirely dominated by the length of the polymer chains. Longer the better. Not too long though, because we can't show it through the hole of the tube that we're looking at. So one of the questions is we see lots of variation in the flow properties of silk. And so is it just the morphs inside of different lengths? Well, there was a suggestion about 40 years ago now that there is a little bit of polydispersity for proteins. What we found that there wasn't, because that's one other really, really interesting thing about nature. Because the way in which proteins are produced, using DNA to template RNA to template mRNA to actually then bring about the production of your proteins, nature will make you exactly the same length polymer every single time, always right, because it follows the instructions on a per unit basis. There's no uncontrolled catalysis going on there, no sort of hopefully trying to stop the reaction before it goes askew. It will produce exactly the same material in a super consistent manner. A lot of the variation that we find in silk and in silk fiber properties and business brush, I think natural materials are somewhat tarred with in terms of, oh, they're a bit variable, not too sure about that. In my opinion, they're not variable. They're incredibly smart. They're very, very smart materials. They're so smart, they will respond to the slightest fluctuation in their environment. They will respond to whiff of acid, they will respond to a change in humidity, they will respond to a change in temperature. The problem is, from an experimental perspective, we're not very good at controlling all of those environmental things when we're doing testing. So I think historically, a lot of the variation we've seen in natural materials is because we're not very good at controlling the testing environment, because we assume they're not as smart as we give them credit. What we found with this is that there's variation in the silk's mechanical properties, but it's nothing to do with the length of the protein. So the other thing that a polymer scientist would say, oh, the concentration, it's the amount of proteins that we have in the gel that will affect its mechanical properties. And we see this huge variation, about 15% in terms of solids concentration, but we find that natural spinning concentrations apparently have very, very little effect. We would expect to the power 3.4, basically, concentration, effect on the viscosity of standard polymer solution. We don't see that at all with silk. So silk basically seems to be a bit indifferent to changes in its concentration. Partly, I think, it's because it's so long, really long molecule, has lots of interactions with other ones around it. So it doesn't seem to have much of an effect on concentration. So if we ruled out molecular weight and we ruled out concentration, that leaves us with quite a few dials sort of removed from an industrial processing perspective. So we have to start looking towards nature. This is where we had quite an interesting observation. So when I first started my PhD, it first off took me like basically a month to grow these silkworms in like 2 or 3 hours a day looking after them, really high up to look after them. And then when you finally get the sample, just depending on how you move it from your Petri dish on your bench onto your rheometer, you can induce just the slightest pinch of your forceps 100-fold difference in mechanical properties. Yeah, so this stuff is a nightmare to handle. It's a nightmare to handle because it will take any introduction of energy, any introduction of flow, and use it for good, i.e. turn it into a solid. It wants to be spun. It has this molecular anticipation that it wants to undergo that phase transition, the smallest input of energy, which makes it a nightmare to handle, basically. So it took a very long time to get consistency. And so this silk feedstock behavior seemed really variable. And even when we got, I would say, good at it, we would still have a viscosity from 100 Pascal seconds up to 5,000, a big variation. But, you know, these proteins are exactly the same length every single time. We shouldn't get this variation. And so we started to think, well, when are we getting this silk? We always took silk from the animal when it was spun, but maybe depending on the stage at which it was spinning a cocoon around itself would have an impact on our results. And so we started to even get down to the minutiae of when is the animal making this cocoon. And we saw that there was a systematic decrease. As soon as we started recording how far along in terms of the production of the cocoon, where we then tested the samples, we saw this systematic decrease. A systematic decrease in viscosity. So we started to look at the metal ions present in the unspun silk as we tested it. And what we saw was a very, very consistent start. Early cocoon, early spinning, late cocoon, nearly finished. We find that calcium's basically exactly the same, magnesium's basically exactly the same, but potassium increases as the silkworm starts spinning more and more and more. We get this increase in potassium ions that seem to be linked to a reduction in the viscosity. That's a chemical way of changing the flow properties. But we tested that, we saw it passively in terms of that's what we observed. We then started adding metal ions in to see if it changed the flow properties of the silk. And what we found is that we add potassium and it makes it more flowable. It also promotes spinning conditions. It makes it easier to transform from a liquid into a solid. It allows for alignment of the fibers. Calcium basically promotes storage. So that original high level of calcium promotes storage, and it didn't really spin when we started to flow it. Lithium prevents aggregation. So that was an interesting side that we went into that I can get into a little bit later. But ultimately what we found is that just by changing the metal ions, we were able to change the flow responsiveness of the silk. Now that's particularly important because if you think about this silkworm excretes the silk into a sac. This sac is then technically outside of its body. So the silkworm can't suddenly start pulling silk back in, changing it and popping it back out. The silkworm doesn't have a furnace inside it where it can start to change the temperature. We can't really change the concentration too much. We pump a little bit of water in, but we know concentration doesn't affect the flow properties. So what dials has it got? It can maybe start to put in some other molecules that would change the flow properties of the silk. But this is a big, entangled mass of molecular spaghetti, which would take ages for stuff of any length to start diffusing through it. But something that could get through it, that could change the flow properties really quickly, maybe a metal ion, could diffuse really quickly and start changing the properties. So we think that metal ions are a particularly useful tool that the animal can use to change its flow properties. This led us into talking with some friends at York University, a chap called Tom McLeish, and his postdoc at the time, now an independent research fellow, a chap called Charlie Schaefer, and they started to model this. And these guys are experts in classical polymer rheology, soft condensed matter polymer modeling. But they were really interested in silk, and they started to predict the properties of silk using this concept of sticky reptation. Now, I'm aware for the next couple of slides, I may lose a few of you. I have to pay attention myself when I'm delivering these slides, but we'll get to the main message at the end of this in a sec. But the idea is that these silk proteins have carboxylate groups on them, where basically your oxygen atoms between two chains can be bridged with calcium as a salt. So calcium is a divalent metal ion, so it can make a divalent metal ion bridge between two chains. Bridge between two chains is molecular friction. Viscosity is friction caused in terms of the amount of force required chains to move over each other. So throughout the silk's sequence are sticky points called stickers, and the calcium bridges between those sticky points, increases the friction inside the gel, which will increase its viscosity. But when the animal wants to start spinning it, what it can do is that it can start popping in some potassium, a monovalent ion, which eats up all the stickers, stops the divalent ions, shrouds the silk molecules, and prevents them from interacting with each other, loosening it all up, reducing the viscosity and allowing the silk to be spun. And that is a concept known as sticky reptation, basically, a additional molecular interaction that makes the polymer physics and the flow properties itself. This is a natural example. There's very, very few examples of sticky reptation in general. This is the kind of stuff that polymer scientists dream of, because if you can functionalize your polymers to have additional interactions or molecular switches that you can turn on and off to change their properties, would be absolutely fascinating and useful. Biology does this all day, every day. They're also able to take this as a concept and predict our biological data. Our experimental data can be predicted through their models using this. And that's really starting to show the benefit of working with modelers, is that they can generate new hypotheses and tweak your experimental data so that you've got an idea of explaining the system. Not only will they change the interactions between proteins, But what happens when you start to flow these sticky molecules? The hypothesis is that the more stickers that you have should help all of these proteins align. And when stretched, they should all align together. All those Lego studs will help them snap together, and we get that amazing hierarchy that developing. The result is a bit surprising. At very low flow rates, we don't get we get these stickers on here, and at low flow rates, they're stretched out, and the stickers that are still present will allow them to interact with the other chains. Yeah, so you can almost imagine them a bit like magnets, where they're just sort of aligning those chains to one another. And then when you really ramp up the stress and pull them and remove that water from around them, they'll snap together. So we think that this stickiness is the registration of the material. But at higher flow rates, unfortunately, we start to see this wider distribution of stretch, which kind of doesn't make sense. We want everything to be nice and even. At sufficiently high rates, we observe that the stretch distribution converges to a steady distribution with independent flow rate, and sticky chemistry is the main control parameter. But what this really means is that they do far more than just get everything all aligned. This idea of sticky reptation creates a distribution. It creates an inequality amongst silk proteins. Some are stretched more than others. The modeling is showing that at very, very low flow rates with no stickers, we can have some silk proteins that are immensely stretched compared to others. Now, I said earlier, wouldn't it be nice if everything was nice and even, nice and consistent? Why would we want this inequity? Why would we want this unusual distribution of stretch in our silk proteins? Well, I mentioned this phase transition. This phase transition undergoes this movement from a solid into, from a liquid into a solid, but it needs nuclei. It needs something to kick it off. And the lovely thing about this, from a polymer flow-induced crystallization perspective, is that these stickers generate nuclei. The super-duper stretched ones shed their water and will start to template others. And that is why we're able to get some energy efficiency into silk flowing, because it will automatically start seeding itself because of some chains being stretched a lot more than others, which will allow them to pop out of shape and act as landing zones for others to sit on top. I'm going to zip through some of these current work slides because I think it's going to be a bit more fun for us to talk about some of the applications of silk. We asked a brilliant question a couple of years ago about whether silk is squeezed or pulled from the animal. Now, people kind of think, oh, spiders are just going to squeeze and the silk will suddenly issue forth. But if I was to give you a piece of chewing gum, and I would say, make me a fibre from chewing gum, you don't spit it out of your mouth. You grab one end, don't you? And you pull it. And that controlled extensional flow, that protrusion allows you to form the fibre. The other question is, silk is quite a thick, dense gel. So what is the force required to squeeze a silk out of a tube equivalent to a spider silk? Here we basically took our tensile tester, popped a syringe on it, and measured the force versus the deformation to get a flow rate, and turned it essentially into a capillary rheometer to work out the force required to squeeze silk out of a tube. And this is one of my favorite bits of a graph. We've got spinning speed in meters per second of a bit of fiber, and we have pressure on this axis. And all of our measurements and all of the theoretical calculations that are out there, you have this relationship between pressure and spinning speed. But when we look at nature, these are the natural spinning speeds. So basically, you have to put a lot of pressure into this, and it will still flow quite slowly. However, there's an upper limit known as the rupture pressure. This is literally the pressure in which the spider would pop if it should generate those forces inside it. It wasn't measured by like pumping up spiders and causing them to explode. It's actually measured from measurements that butterflies use to eggs come out of their chrysalis and pop out. So basically, we're 5 orders of magnitude off to generate the same kind of pressures inside the animal which we squeezed out. But we took a rheometer, we attached a silkworm, we saw a rheometer is basically a very, very controlled spindle that can measure the force and the speed at which you pull something out. We know the cross-sectional area of that fibre, and therefore we can measure the pressure, the forces required to pull silk out of a silkworm. And we found out it's perfectly possible to pull silk, and the forces required are achievable, but the forces to squeeze silk isn't. So it's not extrusion, it's pultrusion. And so if anyone says spiders squeeze out silk, you can say no, they would pop. And silk is mostly pulled from the animal. We've done some more recent work by pulling silk fibers and understanding their mechanical properties. We've looked at changing humidity as we draw silk fibers into from the goo into the solid fiber. We find it can be done, provided it's not too wet. You can actually draw fibers just through extensional rheology. The fibers themselves are rubbish. If you take some learnings from fly fishermen who originally, before the nylons were invented, used silk guts. These were silkworm glands that were dipped in vinegar and then pulled through the teeth in order to make fibers for their fly fishing lines. The trick there is not just the pulling through the teeth, it is the exposure to acid, to vinegar. Because if we look inside the animal, what's been found by collaborators is that there is this steady decrease in pH and increasing acidity as silk moves down this duct. So that's what we did. We wafted a little bit of silk around, a little bit of acetic acid, chip vinegar, around the silk as we were pulling it into a fibre, and it made a massive difference. So this is without any acid, and then as soon as we expose it to acid, all the chains start to align, and it creates mechanical properties that are very, very similar to nature. Not only that, we were able to compare the amount of work put into forming that fibre. And what we found is that the mechanical properties of our ex vivo spun, our artificially spun natural silks, depend on the amount of work input that you put into it. So if the spider's working really hard and pulling it out and sweating, it's making a good silk. And we can start to relate the rate of work input to fiber properties, which is super exciting because that's animal behavior. Animals change their behavior, their spinning behavior, to modify the properties of the fibers that come out of their body. And that depends on how silk self-assembles. So with the last couple of minutes, I'm just going to dive into medical applications because I've sort of said that, oh, we're never going to be able to get commercial silk fibers from sort of bugs and things like that in the short-term future. But that doesn't mean that silk isn't useful for a range of other applications. Because silk is a protein, silk can be used inside the body, and it's been used for thousands of years. It started off from Claudius Galenius in 150 AD, who used silk fibers to repair a gladiator's broken Achilles tendon. You then move on 510 years ago, Ambrose Paré, who did vascular surgery and literally used silk fibers to tie up arteries inside somebody. This was 500 years ago, doing vascular surgery with silk. Absolutely amazing. An innovation, realizing that silk could be unspun and turned into its liquid form and then recast into sponges, films, gels, the whole lot in the 1980s, really opened up this field to a whole range of different types of products that are available on the market today. And today, we find that silk can be reprocessed. It's more than just a fiber. It can be transformed into artificial cartilage. You can make holograms, lasers, disposable membranes, contact lenses, bone. There's not a single part of the human body that hasn't had it replicated using silk as a scaffold. And they're not all fully realized as commercial products, but there are a couple of examples where people are moving forward with this one. A recent case study is Neurotex, which is looking at peripheral nerve repair. where you get yourself damaged. Classic is sort of like, especially hip operations. They have to sometimes cut the nerve, well, they promise you they won't do it, but if they do accidentally cut a nerve, it's one of the longest cells in the body. It goes from basically the base of your spine to the tip of your toe. That is a single cell. If that gets cut, you can imagine sort of live wires, and they're finding quite difficult for them to match back together and heal inside your body. And peripheral nerve injury is very, very difficult to fix. Normally you just leave it and hope that the nerves find their respective ends. But what you can also do is you can make a little straw out of silk, and then you poke it full of silk fibers. And it turns out that neurons, and axons specifically, love growing along silk fibers. It's like a little highway for them. And they'll sort of quite easily use this as a guide, a neural guide, to get one broken part of the nerve to another. And they've done this, and it's been tested in rats. It's really, really helping. I think this is in sheep at the moment. Christina Lacker in general is looking at this. So silk can help with peripheral nerve injury. It can also be used for orthopedics, specifically knee meniscal implants. So one of the problems is we've got a sponge in our knee, it's the meniscus, and cartilage doesn't have a blood supply, which means that if it gets damaged, it takes ages to heal, if at all. And so classic sort of footballer's injury is that you end up with a tear, a massive deformation of the sponge inside your knee, which causes a tear in it. What normally happens is that you tear that, you go to the doctor and you say, this really hurts. And they say, there's not much we can do about it. They give you a steroid injection. And you go back another 6 months later, it really, really hurts. We'll give you another steroid injection. We go back in about a year saying this is almost unbearable. Okay, well, we're going to do a meniscectomy. We're going to just take the sponge out. That's okay for a couple of years until basically the bone starts to grind on the other bit of bone, which is arthritis. And they end up moving you towards a total knee replacement, where they have to solve the problem by a metal ceramic solution. So really, once you're on that road, there's no sort of going back, unfortunately. They saw this as a gap in the market because they thought, silk's got really good properties. Can we recreate cartilage's spongy properties using silk? And it turns out that they can match the mechanical properties really, really well, which means that the surgeons can go in with keyhole surgery and literally just put a little wedge of silkworm-based silk sponge into the cartilage and help basically give a time for the cartilage to slowly repair, but still matching those mechanical properties. It's got really good preclinical data. I found out only last week when my former PhD supervisor, Fritz, was involved in this company. There's a couple of people in Bristol genuinely walking around with silky knees at the moment, and they seem quite happy with it. So I'm really, really excited because this could be one of the very first proper uses of silk in a medical perspective. We're not talking about sort of a coating for breast implant or anything like that, which is what Alden ended up using silk for, because they couldn't use it for anything else. This is a really, really good flagship example, because if this works, it validates our theory, which would be fantastic. So I'm going to sort of leave up on that understanding natural spinning can reveal silk self-assembly mechanisms, but it's inspiration. So next time you look at a spider web, and a couple of days ago, it was brilliant. That frost, you could see all those spider webs in your garden, absolutely marvelous. A material that performs in minus degrees with water on it, but still holds all that mass of water. Fascinating material. We can learn a lot about the way in which we can improve our materials with it. I'm going to signpost you. If you want to learn a little bit more, we have an iron-free natural materials group where we have monthly webinars. They're free. You're more than welcome to join us. We have some really, really interesting talks. We've been running this ever since sort of COVID times now. So we've had, hundreds of speakers come through. They're 1 hour long, 4 speakers, enthusiastic individuals that try and sort of convince you that natural materials are the way. We've got one coming up in February, 16th of February, nice lunchtime. We've got Parvez and Ricardo talking a focus on about natural materials in sport. But they are very varied, the themes, and there are recordings back on the IM3 website if you would like to have a look at any of those. But with that, I'd like to thank, obviously, organisers. This is my group. We've got a couple of people here. This is after the commune over Christmas. If you'd like to learn more, I'm more than happy to answer your questions. Thank you very much for your time.